chief of hematology oncology as well as medical director of oncology here, and he works a lot with the residents. So I'm going to let him take up all his time because Gary told me a lot of slides when he wants to get through. So go ahead, sir.
Thank you. Thanks. I'm no expert in malaria, and I'm not the local expert clinically in sickle cell disease either, so I should probably sit down. This article, which was published recently in the New England Journal, caught my attention and was the impetus for a journal club that I gave to our division a few weeks ago. The reason it caught my attention is not just the clinical impact of this trial, which I'll get into in a little bit, but I have a personal connection with this story, which I'll share with you in a little bit. And this is really a talk about the impact of malaria on the human genome and lessons that those accidents teach us, both in terms of treating malaria and possibly sickle cell disease. This is a slightly dated slide from the World Health Organization looking at cases of malaria by country. And you can see that malaria is widespread in the world with the heaviest impact in Africa, but it extends through Asia and South America. And you can see at the bottom of the slide the cases per 100,000. So down at the bottom, we're talking about 25,000 cases per 100,000, a full 25% of the population affected in some areas in sub-Saharan Africa. The story is a little bit different when you talk about death from malaria. And if you draw this map instead in terms of the impact, in terms of death rate, you see that a hugely disproportionate share of deaths from malaria are borne by the African continent. And this is largely because this is an area that is affected by a number of different types of, this is an area that's affected by many species of malaria, but in particular falciparum malaria. About 1 million people a year die of malaria, and a full 80% of them are children, young children. And the highest death rate from malaria is in fact infants and pregnant women. For those of us who don't think about this, this is a slide that illustrates the life cycle of malaria. Malaria is injected as sporozoites into the bloodstream by a mosquito that's feeding. The sporozoites enter the liver and develop within liver cells, exit as merozoites, and then enter the erythrocytic phase. And it's during that period of time that a great deal of the pathology occurs. It's in the erythrocytic phase that the infected cell matures, acquires different phenotype that leads to adhesion to endothelium. And of course, a percentage of these cells then mature into gametocytes, and the process repeats itself. On a blood smear, this is what falciparum malaria looks like, and these bumps on the surface of this erythrocyte are examples of some of the changes that occur following the erythroid parasitization of the cell. And by EM, this is what happens. These are called trophozoites at this point, and they acquire knobs, and the knobs mediate adhesion to endothelium. And this is where my connection to the story actually started back when I was a first or second year assistant professor in New York. I was studying a molecule in the lab called thrombospondin, which is a platelet protein, had to do with adhesion, and frankly we didn't know a whole lot about it. And in 1986, the sequence of thrombospondin was published. And a homology with malaria was noted. So thrombospondin has so-called malarial circumsporozoite repeats in it. And in fact, this highly conserved sequence was found in thrombospondin, prepared in sporozoites, and various blood stages of malaria. And at the time, I knew somebody who was working in a lab about a mile and a half south of us. Victor and Ruth Nussensweig are investigators who really have devoted their lives to parasites and trying to develop a vaccine to malaria. And I called them and said, what is going on? Why does thrombospondin have this repeat? And Victor Nussensweig met with me and introduced me to somebody else in their department who was working on malaria, who had assembled a panel of antibodies that blocked cytoadhesion of parasitized erythrocytes to endothelium and to cells that were like endothelium. And we reasoned that, well, if this is an antibody that blocks binding, and if thrombospondin is responsible for the binding, maybe this antibody identifies a receptor for thrombospondin. And that was, in fact, what led us to identify a protein called CD36 as a thrombospondin receptor on endothelial cells and platelets. And with John Barnwell, a year or two later, we identified CD36 really as the receptor, the cytoadherence ligand for falciparum-infected erythrocytes. So in this experiment, John basically infected erythrocytes with malaria, and only a certain percentage of the erythrocytes got parasitized. But what this figure shows is that only the parasitized erythrocytes bind to a surface that was coated with purified CD36, the receptor. And we identified this thrombospondin sequence motif, which is in fact responsible for thrombospondin binding to CD36. And some years later, an investigator by the name of Howard showed that CD36 expression on cells, in fact, bound to a specific thrombospondin-like repeat in a surface complex called Plasmodium falciparum endothelial cell membrane protein 1. And so this recombinant protein actually is the immunogen that's been developed as the vaccine. So here's again the life cycle. A blood meal by the mosquito enters the bloodstream, gets to the liver. Merozoites have to enter the erythrocyte, and we'll get later in the talk to the mechanism by which that occurs. There's a maturation process where you get development of the parasite within the erythroid cell. And the parasite has to do a couple of funny things. I mean, the erythrocyte doesn't excrete proteins. And so the parasite has to encode for an entire mechanism to get proteins excreted to the cell surface. And that's what these are called. These are Moore's clefts, and they're actually they're endoplasmic reticulum-like structures that are encoded entirely by the parasite. And they allow the parasite to express on the surface these proteins that include this PFEMP1 that form the structural basis of these knobs. And this is what we now understand from from people who've studied this, that this is in fact one of these knob structures. It's intimately connected with the cytoskeleton of the red cell, actin and spectrin. It's got multiple repeats. One of these is the thrombospondin-like motif shown here in yellow or orange that interacts with CD36. But there's really a very complex interaction of this knob structure with a host of epithelial, endothelial cell proteins that include integrins, VCAM, thrombospondins, selectins, whole bunch of proteins. But a very constant interaction is, in fact, this interaction between this repeat and CD36 across a variety of stages of the parasite. Despite the fact that there's a lot of variation in the genetics of the antigens that are expressed on the surface, it turns out that from a three-dimensional structure standpoint, even though the sequence changes, the structure remains roughly the same, so that the molecular interactions remain the same. And this interaction and cytoadhesion plays a role in the pathophysiology of placental malaria, leading to the high rate of fetal loss and death in women with malaria, cerebral malaria or cytoadhesion in the brain, and microvascular adhesion elsewhere, causing all kinds of difficulties for the host. And there's been an interesting debate in terms of the strategy for developing vaccines for malaria. So if you were the Department of Defense and you were going to send a soldier into an endemic area, you'd like to prevent malaria entirely. And that really means having a vaccine that works against the sporozoite. But that is you have to get 100% of the sporozoites, otherwise they get to the liver and you have malaria. If you were thinking in terms of public health, you might in fact be interested in erythrocytic stages or stages that affect the entry of the parasite into the red cell. And all of the stages of this have been studied. So there have been sporozoite vaccines and other vaccines that have had some varying success. The accident of the constants of the interaction of this repeat sequence with CD36 may be responsible for the early success of this vaccine trial. So again, this was published recently in the New England Journal. It's an amazing study. They enrolled many more than this, as you'll see in a second, but 6,000 children between 5 and 17 months who received vaccines for malaria, and they could receive a total of, I think, 3 vaccines, and there were control vaccines here that included rabies vaccine and one other control vaccine, and the children were monitored for severe malaria or malarial episodes that required care. And again, a total of 17,258 children have been assessed for eligibility. 15,000 ultimately underwent randomization, and we don't have all of the data from these. What I'm going to show you in the next slide is just the cumulative incidence of first or only episodes in the older age category from 5 to 17 months. And you can see here that there's a significant effect of the vaccine decreasing the incidence of malaria in those who receive the vaccine, and including those who just the intention to treat population. So this early success is really remarkable, and I remind you again that the success here, I'll go back, the efficacy of this vaccine is estimated to be about 50% protection in terms of incidence of malaria in this population. And just do the math. You know, there are 1 million deaths per year. Most of them are due to, or a large percentage of them are due to falciparum. And in this age group, this represents a huge potential contribution to medicine. And this is a timeline that was published that shows the development of this vaccine, starting from the first demonstration of the safety and antibody response back in 1995, really a 20-year history of iterative development of the vaccine that culminated in this trial, and the results of this trial that we expect in 2012 and 2014 are long-term results in 2014 in that large group of patients that have been immunized. So this is really the first effective vaccine against a parasitic disease. It targets these thrombospondin-like malarial repeats in PFE-MP1 that's responsible for adhesion to endothelium. There's a 50% or 55% protection against all malarial episodes, 35% against severe, and the death rate in the trial was really too low to demonstrate a decrease in mortality. It's estimated that it's about as safe as a rabies vaccine. There were some serious side effects that I'm not going to go into. These are two patients with similar x-rays. And the point is, so one of them has sickle cell disease and one of them has malaria. I can't tell the difference, and I challenge any of you to tell us who's who here. One has to ask what's in it for the parasite. So, you know, the parasite, the smartest parasite is the parasite that's successful but doesn't kill its host. It's been said that there's been no greater evolutionary force in recent times other than malaria. So malaria has been with the human population since about 10,000 years ago. And in that time, it's exerted a profound effect, and much of what we're going to talk about is just that. So what's in it for the parasite? Why is cytoadhesion advantageous to the parasite? Theoretically, the advantage is that the parasite escapes detection in the spleen. So by adhering to endothelium, it avoids passage in the spleen and is more successful. So here's a map of evolutionary pressure. This is a map of malaria on the left, and the incidence of sickle hemoglobin on the right, and you can see that there's a match here. Sickle cell hemoglobin evolved several times in several places as a response to the enormous pressure that malaria puts on a population. And it's, again, about 10,000 years ago when people began to live in communal settings is thought to be when the malarial parasite entered the human population from other hosts. So there are other types of malaria that clearly affected other organisms and then entered the human organisms. And so this selective pressure demonstrated here is pretty profound. And there's a tough evolutionary choice. So we can have hemoglobin A and be at high risk of malaria, or we can have hemoglobin S, and that's clearly not a selective advantage, high death rate from sickle disease. But the heterozygous state it has an evolutionary advantage and persists, and it's that advantage that leads to the prevalence of sickle cell disease in some areas where there's historically high malaria incidence, prevalence of 20% or higher of sickle cell gene trait. But it's not just sickle cell disease. So pyruvate kinase deficiency, which affects the cell's ability to handle oxidative stress and presumably has other effects on the fitness of the cell and the ability of the parasite to survive, also reduces the parasitemia. So this is pyruvate kinase deficiency showing that the parasitemia in red cells that carry that defect is much less. And so there's been a coevolution of malaria in humans. And this slide is meant to show that. So this is host fitness, and you can see that with increased episodes of selective pressure, different mutations have evolved: sickle cell trait, G6PD deficiency, etc. And this isn't meant to be a literal time scale. It's just the fact that many of the benign conditions, benign red cell conditions that we see in hematology are really the result of this selective pressure. And this panel illustrates some of that. So hemoglobinopathy, sickle cell trait, which is what we're talking about, but also alpha thal, beta thal, hemoglobin C, hemoglobin E, all arising independently as a response to this selective pressure. Red cell membrane abnormalities such as spherocytosis and elliptocytosis, blood group O, because the entry to the red cell is thought to be in part due to glycophorins, and so the blood group O as a selective advantage also, and complement receptor on the red cell, which is also thought to be the receptor responsible primarily for entry of falciparum malaria and polymorphisms in the complement receptor, have evolved. And then, as I said, G6PD deficiency or pyruvate kinase deficiency, all as a result of the selective pressure that malaria puts on this. So how does that work? I mean, what exactly is going on? Why is sickle cell trait or hemoglobin C disease advantageous? How does it protect against malaria? That's really been a question. And there's a very recent report that describes the mechanism here, and it's pretty neat. So, and the title says it really, that hemoglobins S and C interfere with actin remodeling in the Plasmodium falciparum infected erythrocyte. So again, as I showed you before, what the parasite's got to do is it's got to get these proteins to the surface and form these knobs, and in so doing, it's got to do this by making these secretory granules and getting those to the surface of the cell. And so normally what happens is that you get, and this is an electron tomographic image of the picture, the cartoon I just showed you, and so this is the so-called Mohr's clefts, and you can see that these granules are associated with the cytoskeleton, and these granules are what carry the parasite, the PFEMP, to the surface and make the knobs. And this is in a regular normal cell, but in cells that carry SC or hemoglobin C, there's the total disconnection of these Mohr's clefts to the cell surface, so they can't mature, they can't express these proteins on the cell surface. And so what that paper showed is that falciparum encodes its own secretory pathway to facilitate the expression of these knobs. The hemoglobin C and SC disrupt the cytoskeletal attachment of the knobs. and raises the question of, well, why? How does that exactly work? In data that I'm not going to show you, they hypothesize mostly that it's the oxidized hemoglobin or superoxidized hemoglobin, Fe+4, that might be the mechanism. And that ties it together with some of the other enzymatic defects that presumably protect also against malaria. So the mechanism for G6PD and pyruvate kinase may ultimately be similar to this in the sense that there's hemoglobin that becomes superoxidized in those cells. Well, there are other receptors on the cell surface, and this is taken from a review by Leslie Parisi, who is interested in sickle cell disease and identifying receptors that may be activated in sickle cell disease that lead to the similar cytoadhesion that we saw in the image I showed you of somebody with chest syndrome and cytoadhesion and vasculature in the lungs. And so some of the pathways that are thought to be involved is activation of a G-coupled protein receptor and a tyrosine kinase pathway that leads to increased adhesion through an integrin to immobilized thrombospondin. And this is a pathway that's been studied in sickle cell and presumably plays a role in the cytoadhesion and vaso-occlusive disease that we see there. And so I've sort of superimposed cell adhesion in sickle cell and in malaria because they're really quite similar. This is sickle cell down here with some of the players not well illustrated, but as I showed you, some of the same players here as here. So we have activation of pathways that lead to adhesion through ICAM, thrombospondin, CD36, and some of the same pathways identified in sickle cell adhesion to the vasculature. There's an inflammatory cycle in malaria, and that has to do with the rupture of these cells, the release of arginase, decrease in nitric oxide, and a whole inflammatory pathway that ultimately leads to endothelial cell activation, increased adhesion, and increased cytoadhesive events, and ultimately both pathophysiology of severe malaria. Similar pathway is seen in sickle cell disease, where you have intravascular hemolysis, decreased nitric oxide, and the same sort of pathway leading to hemostatic activation. And so the nitric oxide pathway has actually been a target of therapy both for sickle cell disease, not terribly successfully, and in severe malaria for these reasons. There's a very recent paper that ties together the hemolytic events of malaria and sickle cell with some of the immune pathways. And this paper shows that sickle cells generally activate heme oxygenase, and that heme oxygenase is responsible for inhibiting the effect of hemoglobin to produce cerebral malaria. And the malarial parasite induces a tolerance that's manifested through cytotoxic T cells that induces tolerance to Plasmodium infection. And again, this is an example of the parasite using sickle cell as a pathway to evade immune detection. These epinephrine pathways that I showed you on a slide back with that Leslie Parisi talked about is another interesting pathway that relates to both pathophysiology of malaria and treatment in sickle cell. So this is a beta-adrenergic receptor leading to G-coupled protein activation, a cyclic AMP pathway that leads to activation of epinephrine adhesion receptors, and that's the part of a basic pathway that's thought to play a role in sickle cell disease. And there are subpopulations of cells in patients with sickle cell disease who express activation and increased responsiveness to epinephrine. This is malaria, and malaria's got to invade the erythrocyte, and it turns out that invasion is dependent on the activation of a G-coupled pathway. And inhibitors of that G-coupled receptor pathway totally inhibit the parasitization of these erythroid cells. This pathway has been investigated, and the paper that I'm referring to is a paper by Zhang and colleagues, and it's entitled Detrimental Effects of Adenosine Signaling in Sickle Cell Disease. And really, this cartoon at the bottom right shows the pathway that's thought to play a role here. So hypoxia leads to increased signaling through this pathway, increased 2,3-DPG, and therefore more deoxyhemoglobin, more sickling, more hemolysis, tissue damage, ATP release, more adenosine, and there's a pathway. And in fact, pegylated adenosine deaminase or inhibitors to the receptor may in fact be therapeutic targets for treating sickle cell disease. And one of our fellows, Jun Zhang, has been involved in studies with this group, has provided them with patients for these studies, and is a co-author on a paper that extends this observation and shows that in addition to 2,3-DPG, sphingosine kinase 1 leads to the production of sphingosine 1-phosphate that together with 2,3-DPG is actually critical for sickling. And she's a co-author on a presentation at ASH this coming week that basically is summarized here. In the last couple of slides, I'd like to just show you where the field is going in terms of other therapies. Now, this is in mice, but it's a remarkable observation. So this is a paper entitled "Correction of Sickle Cell Disease in Adult Mice by Interference with Fetal Hemoglobin Silencing." So again, here's what happens normally during development. We start off with embryonic hemoglobin, hemoglobin F, contributed by alpha chains from chromosome 16 or beta chains from chromosome 11. And at birth, this switches, and we produce predominantly hemoglobin A, and hemoglobin F gets shut off. And the problem here is that for those who carry a mutation in their beta chain, such as sickle cell disease or hemoglobin C or hemoglobin E, this switch is what brings on the disease. And we know, in fact, that in those patients who have evolved mechanisms to preserve expression of hemoglobin F, the severity of sickle cell disease is modulated. And in fact, 10% hemoglobin F is protective, and those patients won't have any of the phenotypic manifestations of sickle cell disease, even if they were homozygous for sickle cell. And if you look at the population distribution of persistence of hemoglobin F, you see that there's a curve here, and the patients who fall at the far end of the curve, who express greater than 5% hemoglobin F, there's a tail. Some patients all the way out here. And a series of studies have led to, and these studies are called genome-wide association studies, and basically what they're doing is they're doing SNPs and doing sequencing to try and identify what genes are different in this population of patients, the patients who have increased hemoglobin F. And these studies led to the identification of a transcription factor called BCL11A that's thought to be associated with hereditary persistence. If you knock this out in mice, you basically see persistence of these embryonic hemoglobins. In other words, hemoglobin F, the persistence of the gamma chain. And if you do a conditional knockout, you show persistent hemoglobin F. This is persistence of hemoglobin F in these mice. But the real question is this a target? In somebody who already has has sickle cell disease, can we knock out this gene or target it, and is that a true therapeutic target for sickle cell disease? And so what they did was cross the knockout mouse, or the conditional knockout mouse, with a model of sickle cell disease to produce a sickle cell mouse that didn't have this transcription factor. So these are the results. Here's a control. Here's the sickle cell mouse, and this is a sickle cell mouse that expresses human cells in human hemoglobin. So it has all the manifestations of sickle cell disease in a mouse. So this is sickle cell mouse blood showing sickled cells, and when you knock out the transcription factor, you ameliorate all of the phenotypic features of sickle cell in the mouse. And there are drugs actually that are synergistic with this effect, and these are drugs that are available. So 5-azacitidine and vorinostat or SAHA are histone deacetylase inhibitors, and in the mice, these drugs had a profound effect. And bring this right back to the clinic, because there are currently trials ongoing looking at vorinostat in patients with sickle cell disease. So this last paper showed that the inhibition of this transcription factor achieved hemoglobin F levels that were well in excess of 15 to 20%. That's really curative of sickle cell disease and raises the possibility that this is a real new therapeutic target. And lastly, I'd like to tell you about somebody else in the division whose research is likely to impact this field. So here's a model of vascular or vasculopathy in sickle cell disease, pointing out really that there's vascular occlusion, and that distal to that you have tissue hypoxia, and with tissue hypoxia, a change in metabolism and decreased pH. And Li Yang in the division is interested in a family of receptors that are G-coupled protein receptors expressed on endothelium that are pH sensing and are likely to play a role in the post-occlusion pathology that occurs in this disease. I think that's likely to be a fruitful area of research for us. And I'm going to stop there.